respiration?
Cellular respiration happens in every cell, plant and animal. Plants make their own glucose. We eat plants. So one way or the other, everybody uses glucose. So glucose comes to the mitochondria. The mitochondria is the powerhouse of the cell. It's got two membranes, a smooth outer membrane and a wrinkled inner membrane. And the glucose shows up, it can't get in, it's way too big. So we go through this first step called glycolysis. Glycolysis means glucose to split. And we use 2 ATPs of energy to break that glucose in half. When it breaks, 4 ATPs are released. And so we really just gained 2 ATPs. And that's the important part of this, is the gaining of ATPs. So now, I can see I don't really have a true piece of glucose here. Okay, now I take this glucose that has been broken, and when it broke, 2 of the H's are lost. They are lost from that big molecule, and the H's are picked up right away by an H acceptor called NAD. NAD picks up an H, and then it's called NADH. NAD picks up the H, it's called NADH. And so now I'll show you what happens with those NADHs later on. But these 2 pieces I have remaining are called pyruvic acid or pyruvate, and they are ready to go into the mitochondria. As they pass through that first membrane of the mitochondria, they go through a transition reaction that modifies them a little bit. An enzyme called acetyl-CoA is used to break these carbons and hydrogens off of that pyruvic acid, and now this acetyl-CoA molecule is left over, and I have to know what to do with these pieces of my original glucose. The hydrogens, they're picked up by NAD, making NADH. The carbons are garbage. They are exhaled as carbon dioxide. And that's it. Now I've got 2 pieces of acetyl-CoA, and they are ready for the citric cycle. In the citric cycle, my acetyl-CoA is broken, 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 broken. All the remaining bonds are broken, and I have to do something with each of those pieces. The carbons, they're the easiest. They're exhaled as waste, carbon dioxide gas. 3 of the H's are picked up by NAD, forming NADH. 1 ATP gets charged. Remember, that's what this is all about. And the last H is picked up by a lower energy H acceptor called FADH, forming FADH2. And it's going to sit lower on the electron transport chain. I go through the citric acid cycle twice because I have 2 pieces of acetyl-CoA. This second piece is broken all apart. 2 carbons exhaled as carbon dioxide gas, 3 H's picked up by NAD, forming NADH, 1 ATP gets charged, and my last H is picked up by a lower energy H acceptor called FADH, forming FADH2. Lastly, I've got the ETC. And remember, our goal here is to charge ATPs. And something I know I forgot to mention earlier, because we're in a hurry, is oxygen is being pushed to the inside of the mitochondria, and it is waiting for all of this to happen. So oxygen wants 2 more bonds, and these H's are just what it's looking for. These H's are going to come through a protein that sits on this inner membrane, and as they come through, each one charges 3 ATPs. So I've got 10 beads times 3 is a total of 30 ATPs that get charged, and as those H's come through, oxygen catches 2 of them, forming a water molecule, which we exhale all the time. These 2 lower energy H's come through those proteins. Each one charges 2 ATPs. 2 times 2 is 4. Oxygen catches them, becoming water, which we exhale. And so just to recap all of the excitement, I gained 2 ATPs in glycolysis, 2 ATPs in the citric cycle, and 34 more ATPs in the ETC, for a total of 38 ATPs from 1 original glucose molecule, just because oxygen was available. This part happens without oxygen, but these require the oxygen.
Oh, okay.